Purpose of photosynthesis. Plants do photosynthesis, but what's the purpose of? Well, children, the purpose of photosynthesis is one reason. It's because we want to convert light energy, right, from the sun into chemical energy. Okay? Basically, well, we're going to take that chemical energy and we're going to change it into glucose, right? Which is a type of sugar that stores the energy. This glucose can be used by the plant for growth, repair, and just normal function, or it can be passed on to other organisms that it eats the plant. And a byproduct of photosynthesis is oxygen. We know plants release oxygen, right? So photosynthesis creates oxygen also, right? But before we start photosynthesis, let's talk about the equation for it. All right, for photosynthesis, we start off with 6 molecules of carbon dioxide. 1 second. 6 molecules of carbon dioxide, of CO2, plus 6 molecules of H2O, right? Plus some light energy from that sun that I've beautifully drawn. Right, and we're going to change that and turn it into glucose, which is C6H12O6. Right, and then as our byproduct, we're going to get oxygen from plants. We know plants give off oxygen, right? So that's our formula for photosynthesis, right? We're going to use this carbon dioxide in the light independent reactions. Okay, independent, that's spelled wrong, but it's okay. Reactions, and then we're going to use H2O for our light dependent reactions. Right, so this is going to happen, this is the Calvin cycle, obviously. And this is going to happen in the thylakoid. Right, as a byproduct, we're going to get this glucose, right? And we're going to get this out of the Calvin cycle, right? From our 2 G3Ps, right? Also, we know that photosynthesis is not just done by plants. We have, in plants, it's done in the chloroplast, right? We have algae that does it, right? This is, even though they're single-celled or multicellular, they still do it. Right? We have cyanobacteria, which, even though they don't have chloroplast, they perform photosynthesis in the cell membranes. Okay, so photosynthesis, like any organism that has pigments like chlorophyll, photosynthesis happens in the chloroplast. So the first part of photosynthesis is light-dependent reactions, which happens in the thylakoid. Here, these two big ellipses are like thylakoid, right? So in the thylakoid, we have a couple of things that happens. We have we have a bunch of proteins in an electron transport chain, which I will draw right here. Okay, now I've drawn the proteins, and now here I have in the middle part the electron transport chain. On the left, I have this protein called PS2, and on here on the right, I have PS1. Okay, and this is another ETC. Okay, so what happens basically is the sun, it comes here, and we take H2O coming from our soil, and what it does is it splits it up into oxygen and hydrogen, okay, and electrons, okay. So now what happens is that we're gonna label these electrons in these red dots. That's right. So we have these electrons, and these electrons will go through here in PS2 and they'll be charged up by the sun. Right? So it's gonna charge them up through here in the electron transport chain. So as we go through here, it goes through this entire chain, okay, and then it goes through here. And as what's happening through this entire thing is that these electrons are slowly losing energy, right? And what that released energy will do, as I will show, is that it will allow for hydrogen to come in. It looks like sad faces. Funny. Okay, so then what happens is that we have this hydrogen that's already outside, right? We have a bunch of hydrogen that's outside, right? And due to this released energy, we're going to take that hydrogen in and we're going to add it inside of the lumen, okay? So inside of the stroma or whatever, inside of the thylakoid, right? And we take this and we put it inside of the thylakoid. Now, as I said before, the electrons, since they're losing energy, they get recharged back up in the PS2 and PS1. And then we have another electron transport chain. So then what happens is, since we have this hydrogen outside, right? We're gonna take all of that and we're gonna make something out of it. So we have hydrogen and we have our electrons also, right? Have our electrons, and then we're gonna use NADP from the Calvin cycle, which we will talk about later, and we're gonna turn this into NADPH. NADPH, which ironically we'll again use in the Calvin cycle, okay? So another thing is that this oxygen, right, it's a byproduct of this reaction, so we released it. And what happens due to the hydrogen, since we have hydrogen on the inside and the hydrogen on the outside, it says since we have it coming outside and inside, right, we have this electrochemical gradient, okay? And essentially what's going to happen is that because of this flow of electrons, since there's also electrons outside, it's going to want to leave, right? So we have this protein called the ATP synthase. And what the ATP synthase will do, which is here, ATP, I'll draw it over there. ATP synthase. It will use this flow of hydrogen coming outside, and it will help put a phosphate back inside of an ADP that we get from the Calvin cycle. So we have an ADP, and we're going to turn that into an ATP, okay? And that's due to that flow of hydrogen coming outside, right? So we're going to add a phosphate group back inside of this, okay? So now we're creating ATP that will be used for our Calvin cycle, and we've also created NADPH, okay? And oxygen. So that's the first part of photosynthesis. Okay, so now that we have our Now that we have our NAD, NADPH, and we have our ATP that we got from the light dependent reactions. We're gonna use those, and we're gonna go to the Calvin cycle. Okay, so the Calvin cycle is your light dependent reactions still happening inside of the chloroplast, but it does not happen inside of the thylakoid, but it occurs in the stroma. The light dependent reactions, they happen in the thylakoid, right? So this is happening in the stroma. So I will draw this Calvin cycle now. Okay, so let's start our Calvin cycle. Okay, so first we start off with some carbon dioxides. Okay, so we have 3 CO2s. So our first step is carbon fixation. Carbon fixation. Okay, so basically what happens is that we're going to take these 3 carbon dioxides, and basically what these we have is a molecule called ribulose biphosphate, right? And this is a 5 carbon molecule. So we're going to take that CO2 that we have, and we're going to take these 5C. That 5C is basically our ribulose biphosphate. And what we're going to do is we're going to take that and make it 3,6C. Okay, so this becomes 3 unstable 6 carbon molecules. Okay, and these 6 carbon molecules immediately split in half, and then they become a 3 carbon compound. Okay, and it's called PGA, or phosphoglycerate. Okay, so because we get 6 of 3C. Okay, now what happens is that we have our NADPH that we had before, right? NADPH, right? We're gonna use that, and that's gonna be a reducing agent, okay? And I'll show you why. Okay, and we're gonna change that into NADP, which we can again recycle back into the light dependent reaction. So number 2, we have reduction. So basically what we're gonna do here is we're gonna take this 6 phosphoglycerates that we have, right, 3 PGAs, and we're gonna convert these into 6 G3Ps. So 6 or 3C, it goes into 6 G3P. Okay, I'm gonna write it again here because it's easier to see. G3P. Okay, one thing that you should know is that we're gonna take out 1 G3P over here, right? So 2 G3Ps is equal to 1 glucose. What does that mean? If we're taking out 1 G3P here in the cycle, right, that means we have to go through the cycle twice to make 1 glucose, right? So converting this phosphoglycerate into G3P, we're going to need ATP here. So we're going to use ATP, and we're going to convert that into ADP. Okay? Right? Now, what happens is we switch it back. We switch this G3Ps back into the 5G3, what is it called? 5 ribophospholates that we had initially, right? We switch it back into 5 ribophospholates. So we have this last step, regeneration, in which we take now we have 5 G3P, okay, and we convert this back into 3 of 5C, or 5C is obviously our ribulose biphosphate. So that's our cycle that we use to turn into to get our glucose, right? So then this way, now that it's back into 5C over here, we can once the cycle again, and when more CO2 enters, we can continue this Calvin cycle, and now we have glucose, right? So in the big picture, this occurs in the stroma. We use our CO2, we use ATP, we use NADPH, right? Outputs, right? We have G3P, which becomes glucose, right? And we have ADP, which goes back in to our light dependent reaction. Then we have NADP, which again also goes back into the light dependent reactions to get charged again, right? So that's one. Talk about enzymes. Enzymes and photosynthesis. Now, enzymes are really important because obviously enzymes speed up the chemical reactions that happen in photosynthesis. Without them, the reactions would happen too slowly to stay in life. So let's look at the Calvin cycle. In the Calvin cycle, one enzyme that we use is ribosquil. Okay, and what it does is it helps attach CO2 to RUBP during carbon fixation. So we have carbon fixation, which we know is the first step, right? And basically what it will do is that when we have the CO2 that we use and we have our RUBP, it helps attach that RUBP to so it helps to attach to the carbon dioxide to convert it into PG. So it'll be easier for it to convert into PGA. That's one use of photosynthesis.